Hello, my name is Elizabeth Levinshteyn. I'm in 8th grade, and this is my work called Testing, Designing an All-Strain Inclusive Test for SARS-CoV-2. Introduction. First detected in Wuhan, China in 2019, COVID-19 became a global pandemic. With 95.4 million cases and 2.03 million deaths worldwide. This data is from Johns Hopkins Coronavirus Research Center and is updated as of January 18, 2021. COVID-19 is a respiratory disease caused by a positive RNA virus called SARS-CoV-2. SARS-CoV-2 is a beta coronavirus similarly to SARS-CoV-1 and MERS. Many scientists believe that it may have evolved from a similar beta coronavirus that is hosted by bats. The SARS-CoV-2 genome codes for 29 proteins, including S protein, E protein, M protein, N protein, and others. 19 of these 29 proteins have unknown functions, and this entire genome is 29,903 base pairs long. In order to properly respond to a global pandemic, it's very important to have a test for the pathogen. About a week after the first confirmed United States case, the CDC developed a test that uses human mucus and respiratory tissue to test for the presence of SARS-CoV-2 in someone. This test has 81.5% to 92.2% sensitivity rate, but it's likely to decrease as more variants of the virus emerge. This test is also highly criticized for having a high false negative rate. The CDC's test uses a process called RT-qPCR, which detects and amplifies viral RNA using primers matching, in this case, the N protein. Then the amplified DNA is measured to see if SARS-CoV-2 is present and in what quantities. Research question and hypothesis. In this study, I was interested in developing a new test for COVID-19 that would detect all known variants of SARS-CoV-2. I hypothesized that different regions of the virus mutate at different rates, and I could use the regions with low mutation rates to design a new test that is more tolerant of SARS-CoV-2 variants. Materials and methods. Sequences of beta coronaviruses were taken from GenBank NCBI. Protein sequences were compared using Clistal Omega. Mutation patterns of 3,819 variants were collected using NextStrain, and the most efficient primers were pulled using Primer 3. Results. So throughout my research process, I had two criteria that I used to pick out a viable region in the SARS-CoV-2 genome and then pick the feasible primer pairs inside that region. The first criteria is sequence similarity. I had to make sure that the region that I chose had a low similarity to other beta coronaviruses so that my test wouldn't accidentally yield a positive result when detecting another beta coronavirus. Another important criteria is mutation rates. I had to make sure that there were low mutation rates between the different SARS-CoV-2 variants in my chosen region so that my test would actually detect all variants of SARS-CoV-2 and none would evade the test. Next, after picking the viable region, I had to pick the primers within the region and then develop my test. The ultimate goal is to identify the most viable region in the SARS-CoV-2 genome and then pick the feasible primer pairs. Protein sequence similarity. I retrieved sequences from GenBank of 19 beta coronaviruses with complete genomes. Then I created phylogenetic trees and sequence alignments. As you can see on the left, this is phylogenetic tree of S protein. However, all of the proteins showed that SARS-1 was actually the most similar to SARS-CoV-2. I only considered proteins shared by all 19 beta coronaviruses to eliminate the possibility of accidentally using a protein that is more likely to mutate and disappear in the future. In addition, you can see in the sequence alignment that with every line being a separate beta coronavirus, there are a lot of similarities between the proteins. I considered proteins NSP1 through NSP16 and S, E, M, and N proteins. NSP2, NSP3, and NSP4 had the lowest similarity. A reason why it's so important to have a test that is sensitive to all variants of SARS-CoV-2 is because, as you can see, different regions from around the world have different clades of the virus that are more common. For example, in the United States, the 20C clade is the most common, while in Australia and Russia, the 20B clade is more common. This is why it's very important to be able to pick up on all variants of SARS-CoV-2. Mutation patterns. So I used next-strain data from 3,819 variant samples of SARS-CoV-2 with mutation patterns from all around the world. SARS-CoV-2 mutates with an annual rate of 21.576 substitutions, which is a mutation rate of 9.22 times 10 to the negative 4 mutations per site annually. This is 4.4 times slower than influenza, meaning that it is possible to develop a test that catches up with all of the mutations. So mutation rates between proteins were compared to help decide which region had the lowest mutation rate. They clearly differ between proteins, as I'll show on the next slide. N protein is the one used in the CDC's test, and it actually has the highest mutation rate, which is most likely why there's so many false negatives with this test, and means that a lot of the case numbers we see are actually a lot higher. So as you can see on top, that is N protein, and on the bottom is NSP10. You can see that the N protein has many more mutations per site than NSP10 does. So these are the mutation rate results. NSP10, NSP12, and NSP8 had lowest mutation rates. I then used the two criteria to make a chart with final protein rankings. As you can see, I ended up choosing NSP3, NSP4, and S as the highest ranking proteins. So RT-qPCR requires primers that are approximately 20 nucleotide long strands of DNA. They bind to the chosen RNA region, and then viral RNA is converted to DNA and amplified through PCR. NSP3, NSP4, and S genes were chosen based on the described criteria. So the thing with primers is they have to be a perfect match to the complementary strand. So that means I had to find a mutation-free region of 20 plus nucleotides. This actually narrowed down the entirety of the 3 genes that I selected to just 32 regions. Next, primers work in pairs, and the optimal distance between them is 70 to 200 nucleotides, which then narrowed down the list to 20 pairs. I used primer 3 and put in sequences of NSP3, NSP4, and S genes as input, and excluded all undesirable regions. Primer 3 yielded 2 viable primers in the S gene and 1 in NSP4. I named them NSP41, S1, and S2, and these are their positions in the genome. So these are my chosen primers on the genome, and you can see there's NSP41, S1, and S2, and S1 and S2 overlap. So next, the explanation of my test. So my test uses NSP41, S1, S2, and RP, which is a human respiratory protein. It will use a human control, a viral control, water control, and then finally the actual clinical sample. So with human control, if the test works correctly, there should be no viral RNA. So NSP41, S1, and S2 should not be present, but RP should. With viral control, it's the opposite, where RP should not be present while all of the viral primers are, and water control should test negative for all of them. If there are any results other than these, they will be deemed inconclusive because the test is likely contaminated. Discussion. In conclusion, I used 2 criteria to pick suitable regions and primers. These 2 criteria were low similarity and low mutation rate. NSP4, NSP3, and S genes were chosen, and they yielded NSP41, S1, and S2 primers. There are different variants of SARS-CoV-2 all around the world, which highlights how important it is to have a test that picks up on all of these variants. N gene, which is used by the CDC test, has the highest mutation rate, which is likely why there are so many false negatives. And periodically redesigning tests may lead to fewer false negatives in the future. Limitations and future directions. Due to my lack of access to a biosafety 4 lab, I had no confirmation or real testing of my test. However, my work is a crucial first step. Future mutations may evade the test, and databases are constantly updated and are limited. And I tried to use the most recent data. Finally, novel tests continue to be developed by other research groups using different proteins, and many of them also yield fewer false negative results. These are my references. Thank you for coming and listening to my project.